have not yet collected your Unit 18 notes on purpose because you need them to study. So on Monday, I will collect 18 and 19 notes together. And I will grade 18 and 19 syllabus statements also on the day of your test. Yes. Oh, no, Miss B, stay safe. Thank you. I am recording. Thank you, guys. Okay, we do need to spend a moment talking about materials. Basically, I know some of you might be waiting to start your experiment because you don't have materials from me yet. I've kind of been waiting to order materials because I don't have a whole lot on our order list yet. So I am ordering materials tomorrow. Which one is it? Which one is it? Here we go. Oh, that's not it. It's one of these. Shoot, I thought I had it on. Don't get too far into those conversations. We're still talking up here. Internal assessment, IA. Under that, I have your IA materials order sheet. Let's take a look. I'm ordering tomorrow. So far, I have orders for a few people. Okay. So I'm ordering tomorrow. I really like how a lot of these people found what you needed and found a link for it. That very much helps me out. I'm definitely going to check your link and make sure it's good to go for what you want, but it really helps to have it there for me. Manny, I think you said you had the links at home, so don't forget to get those in. Okay, so I plan to send this order to Ms. Branca tomorrow to order materials. So if you know what you need to get it on here tonight. Okay. This will not be the only order, but I do want to get you guys your materials so you can start running your experiments. I will probably do another order maybe next Friday, depending on how many people add their stuff to the list. Okay. But ideally, you will have your stuff on here before tomorrow, so you can get your stuff, you can start running your experiments right away. I did say on the agenda, if you need help filling it out, or if you're still not done with your exploration, so you don't even know what you need, you should be coming to enrichment. If your exploration is missing, we're actually going to start sending emails and phone calls home to be expressing to parents that you are lacking on this very important assignment. Okay. Maybe it's not your final draft, but you need to have something in at this point. So we're here during enrichment. You guys need to get in and get that done. All right, let's keep going in our notes. So we are on number 4. What were the first 3 pieces of evidence for evolution? Any order? Yes, we can observe evolution happening, right? So observing it happening in current situations, such as antibiotic resistance, that was one piece of evidence. Huh? Yes, artificial selection. That's basically us forcing evolution. With our smart human brains, we can cause evolution to happen for our specific uses. Yes? Fossil evidence. Thank you. Fossil evidence. Good. So as we already can get the pattern, this whole unit is about evidence for evolution. Okay, so last unit we talked about natural selection and the tenets of evolution, what causes evolution to happen. But what's the evidence? Okay, so we have those 3 you just listed. Evidence 4 is a pretty strong piece of evidence as well, homologous structures. If you read the due now, it did say to get a materials box. So if you don't have one, you need one. Okay. Pretty much all of this is written for you. Right now, you are probably writing down the pentadactyl limb. So for the pentadactyl limb, those are in a color for a reason. You can write it in pencil, and then we can color it a color in a minute. But we will be using those colors. So we have pink, purple, green, blue, red. So at your earliest convenience, you need to get those colors in front of you. So while you're writing, homologous structures. We already know our root word homo means what?
Same.
Same. Good. So homologous, we actually already used that in terms of homologous chromosomes, right? Same chromosomes with the same genes, same traits. Homologous structures is just a little bit bigger. If you look at the picture down below, you can see that we're looking at bone structure for this example. So comparative anatomy is a whole section of science. I actually took a whole class on comparative anatomy, and it was super fascinating. But we just look at the anatomy of different animals and we compare them. Exactly what it sounds like. And through that comparative anatomy, we see that there are homologous structures that exist among different species. What does that mean to us? That means that it implies that there was a common ancestry. Okay, like we looked at the whale. We saw that the whale, although the back legs are very different looking, they are still there. And the only reason, the most sensible reason for those to be there is because of the ancestral legs. Yes. Yeah, you just don't see it because it looks like a fin, but when you zoom into their skeleton, it's fingers. Yeah. If you took off their fin, maybe. But the shape of their fin keeps it solid. And I don't know, we could look at their joints. I don't know if their joints have kind of solidified. We would have to look more specific at that. Okay, do we have our, you should also define pentadactyl limb. So like above your bullet points, what that means. Penta is what number? 5. Pentadactyl limb means there are 5 pieces to it. Okay. So pentadactyl limb is a limb bone structure common to all tetrapods. Whether you see all of the digits or not is one thing. But if we look at the skeleton, we see this homologous structure of a pentadactyl limb. Pentadactyl limb. All right, if you don't yet have those colors, you need to get those colors. Pink, purple, green, blue, and red. If you need 2 boxes, there might be extra boxes in the back. Okay. So, the first one, actually, I'll just give you the coloring. So the first one was the single bone proximal to the body, which we call the humerus. Point to your humerus. Based on this picture, can you figure out what your humerus would be? This one, upper arm bone, we would call it. And as you can see, the whale has one, the frog has one, horse, mice, human, bat, and bird. Different shapes and sizes, but they all have this main bone coming out of the shoulder socket, being the upper arm bone. So you should be coloring that first one in pink or whatever color you think. Well, so we're noticing this homologous structure of this humerus bone. Okay. What's the next bone? The radius. The one of 2 distal bones. So you know how your forearm has 2 bones? The distal and the radius and the ulna. Okay. Notice that in all of these animals, they have both of those bones. These bones allow us to be able to twist our arm, our hand up and down, supinate and pronate our hands. It would be pretty lame if you were just stuck in one hand position for your whole life. Has anybody ever broken one of these bones? Okay, so right now the green and purple go next to each other. So right now on your lower arm bone area, it's green and purple. And blue will be below that.
Do it. That is. That's, oh my gosh, I always think that all the time.
All right. So again, we're noticing those bones are slightly different, right? The whale bones is much shorter and fatter, and the bird and the bat are much longer and skinnier and smaller. But it is still homologous enough to imply common ancestry of a tetrapod. All right, what was the blue ones? The wrists. We call these our carpals. You've heard of carpal tunnel? It happens in your carpals. So, so that's the little section. I know it is kind of hard to tell the difference between purple and blue, but the blue is this little bones right above the red.
Give me the green, Kessler.
Wait, in the horse is the green fused to the purple?
In the horse. Yes, it does look like it is fused, and this does happen. Like we talked about in the hoof. Like for example, when you look at the horse hoof, it does not have 5 digits. But what evolution caused over time is that those 5 digits fused into one. But if you do look at the ancestry of horses, they did start out with 5 digits. And then they slowly fused into the one hoof that they have today. Okay. And then we're done with blue, you go on to the red. My favorite name of bones.
Kessler, let me have it.
Most complex thing.
The phalanges. Series of bones and digits. The phalanges. Look at the bat. They have super long, skinny fingers. So it's interesting when we look at this because if you looked at a bat and you looked at a human, you might not think there's very many similarities.
But there is.
But there is. When we look at the skeleton, it's very similar. Okay. So we are all tetrapods. All of these animals, there's Shino talking. All of these animals are tetrapods. And they all are similar in this homologous structure of the pentadactyl limb. Pretty sure that is a syllabus statement. Yes, 5.1A1. Comparison of the pentadactyl limb of mammals, birds, amphibians, and reptiles with different methods of locomotion. All right, are we done coloring? Yes. We do. You'll have to ask Mr. Tena, but he knows this space somewhere. Yeah, the bridge. Oh, really? Just like fly out. I've always wanted to go see that. The bats. Like you can see it's like this huge like swarm of bats. They like fly out at night. And they all live under that bridge. You'll have to ask Kalia. She lives by it. Field trip. If it happened during school hours, I would say we should definitely go. But okay, moving on. So in the world of homologous structures, I'll wait. In the world of homologous structures, we don't just stop at physical things that we can see like bone structures. Bone structures is very easy for us to study. But there are other homologous structures as well. Molecular. These can be homologous. So we look at the DNA base sequences. We can look at the similarity of the DNA between organisms. You're asking me to write this? I don't think you have to write this slide exactly. Cellular, we can look at similar cellular structures. Structural, we just looked at that, bones, muscles, appearance. And even embryological, same embryological development pathway. If you've ever looked at a human embryo and a chicken embryo, they actually look very similar. Over time, we evolved to look different, but at a very early stage of embryonic development, all mammals look very, very similar. Here's an example of an elephant foot. An elephant foot and a human foot.
Oh, it's the same thing. They have more fat on the bottom.
Yep, they just have this extra huge padding. But if you look at it, it's pretty much exactly the same. Like you see the heel bones here, here.
That scared me.
They even have little toes down here. It's an elephant foot. Homologous structure? Yes. Yeah, it's bigger. These aren't drawn to scale. Yeah, the human foot has been blown up.
Giant foot. Imagine like what people thought back then when we first dug up
A skeleton?
a skeleton.
Yeah.
Whoa!
A giant human. Another homologous structure we are seeing here is tusks. So the beaver and the elephant, they both have tusks that they use. Here, you need your colors again. You have this graph in your notes. All right, we've got two things here. Two things can happen: divergent evolution and convergent evolution. And homologous structures gives us a clue to one of them. So divergent evolution, you're going to circle that in yellow highlighter. We have yellow and orange here. I guess you could choose whatever colors work for you. Huh? Yes. And then you're going to write what's in the yellow box and put a yellow box around it. Basically make your paper look like this paper, except for the good explanation.
Thank you.
So, yes. This doesn't matter. I mostly am just worried about the yellow going together and the orange going together. You don't even have to orange. Pick two colors and make them different. So, if you guys think back to your math skills, you probably have dealt with these words divergent and convergent, right? Have we? Oh, I don't know. Graphs that diverge and graphs that. Yeah. Hopefully you know what these words mean. To diverge is to go away, to converge is to come together. So, with divergent evolution, that's oftentimes kind of like how we think about evolution. We think about species having these mutations, giving them genetic variation, and then becoming more and more different over time. We can still see evidence of the similarities, but they become more and more different over time, right? What also can happen is species can actually become more similar. And that is oftentimes the confusing one. When we think about giving species names and trying to organize how they evolved, convergent evolution makes it tricky because they did not have similar ancestors. But oftentimes they will become convergent because they might be in a similar environment or have similar selective pressures. So they will evolve similar traits, even though they weren't connected through ancestors. to know the difference between these two things. Divergent, they're becoming more separate. Convergent, becoming more similar. So this is connected to our homology. If you look down below a little bit, you'll see that one gives us homologous structures and one gives us analogous structures. You guys know what an analogy is, right? Something that is what? If you were in English class, how would you define analogy?
Or something that
Right. It's not exactly the same thing, but it's a similar thing. Good. A lot of times we use analogies to teach new topics, right? You understand this, so I use that to teach you something new. Okay. Adaptive radiation, please also color that the same color you did divergent. So if you did yellow on the left side, yellow for adaptive, yellow for results in homologous structures. We want to know that divergent evolution, adaptive radiation, and homologous structures all go together. Hence the color coding. So adaptive radiation is basically divergent evolution. Notice at the top, it says divergent evolution equals adaptive radiation. Okay. Radiation is like they're becoming separate, right? For example, the finches. They became more separate because they were in different environments, different niche, different traits. Go ahead. So this results in homologous structures. We can see that they are different, but they all came from a same ancestor. Therefore, they're going to have these homologous structures. Okay. Their beaks all look different, but they all have beaks. We want to make it that simple. If we looked at the skeletons of these birds, they would be very, very similar. If we looked at the DNA sequences of these birds, they would be very, very similar, right? So homologous structures happening there. Now, convergent evolution is where it can get tricky. So, oops, that should be in orange. I don't know why I made that green. It should match your orange or whatever you did on the right-hand side. I think I meant to make my orange green. I'm gonna just make my orange green. Okay, but either way, whatever color you had on the right should all match. So convergent evolution can be tricky. So again, this is where organisms end up looking the same, even though they're not from common ancestry. So our example here is streamlining. So if you notice the shape of these animals to be not aerodynamic, hydrodynamic. In order to be hydrodynamic, they have this streamlined shape of their bodies, right? They have pointy faces and then goes down to a long, skinny, kind of streamlined shape, right? But a penguin is a mammal. Is a shark a mammal? So these two, a penguin and a shark, are not very closely related. They do not have closely related ancestors. But they do end up looking the same because they live in a similar environment where they need to swim fast, right? Penguins need to swim away fast from their predators, and sharks need to swim towards their predators fast. And same with dolphins, right? Dolphins and sharks, we know, are not closely related. Dolphins are mammals. They do a lot of human-like behaviors. Sharks, not so much, but they look the same. So you might want to call these homologous structures. You might want to say this dorsal fin and upper fin, and I forget all the names of the fins. You might want to say those are homologous structures. But if you were to look at the skeleton, they would not be the same. If you were to look at the DNA, it would not be very close. Okay, so it's not homologous, it's not coming from the same ancestor. It's analogous because it's similar but different. Yes, go ahead. I think they are. I don't know exactly the difference. I don't remember. I know they're very similar to dolphins. Okay. Molecular homologies. You have a page on this. Basically, this is us looking at protein and DNA sequences, which are related, right? The DNA sequence is code for the protein sequence. So the more similar the sequence, the more closely related the species are. And vice versa. The less similar the sequence, the less closely related the species are. Okay, I have pictures to show you. But if you remember, I know we didn't finish our rock pocket mouse, but he started to show, remember when they were sitting at the computer and they could see the letters on the screen and he was showing where the similarities and where the differences were? So we have the databases where we can put in a dolphin set of DNA and a shark set of DNA. We can look at, we can line it up, and we can see how similar is it. So you can imagine if I had my DNA and your DNA in this database, they would be, you guys remember the percent? What percent the same? Yeah, like 99.9%. My DNA would look exactly the same as yours, which is crazy that only that 0.1% makes us look so different. But if we were to put a human and maybe a chimp, okay, it still would be 99% the same, meaning that we're very closely related. But if I put me and a potato, hopefully, it'd be very different.
Hopefully.
Remember, we talked about that already, that some organisms that are more simple actually have more genes and all of that crazy stuff. But this is a homology we can look at. If we put human A and human B and they're the same, we would say we have the homologous structure of our DNA. Our DNA would be considered a homology. Okay. The less time has passed, the less time sequences have had to acquire mutations. This is why they are so similar. So when one of the jobs of scientists over time has been to figure out the mystery of exactly how and when certain species evolve. We want this perfect map that tells us all the nodes and all the connections of the story of evolution. And they're getting a lot closer now thanks to these homologies, these DNA and protein homologies. Before, scientists would just kind of go, this animal looks kind of like this animal, so they group them together. But because of our analogous structures, because of our convergent evolution, that picture wasn't very realistic. So we'll look into some organisms that used to be grouped together because they looked like they should be grouped together. And now we know that they're not because we looked at their DNA and their DNA was super different. Okay. 5 seconds? 0 seconds? 5 seconds. I'm going to go forward in time. Okay. So the more recent a common ancestor is, the more closely related the species are. So this will all start to make more sense when we get into cladograms, which is a upcoming unit. We'll look at how the branches and the nodes of the branches kind of give us the story of when was that common ancestor And how far away was it? And the closer it is, the more closely related the species are. The more distant a common ancestor, the less closely related. And then the bottom one is talking about kind of a summary. The protein, if they're closely related, they're going to have similar proteins, similar DNA, and they'll be in a similar location on a phylogenetic tree. So phylogeny and cladograms is not one of my strongest points, but I'm sure with Miss Viste's support, we will all be. Every year I teach it, I understand it more and more, but it can be very complex, even for the smartest people. Okay. I was just checking something. You guys freak out so easily. You guys need to do more yoga.
Yeah, because if we miss something, then we lose points on the past.
If you miss something, you have the presentation on Google Classroom. No excuses, Jaden.
I'm just trying to get this done.
Student of the month or whatever.
Oh, yeah.
Student of the triad.
That's segregation right there.
I expect more from you.
I'm leaving this.
I don't think you can. Your mom works.
Yeah.
You're stuck.
Yes.
Okay. Oh, I can't wait till my son is here. It'll be years in the future. I'll be that mom that. Go ahead. I'll be like checking on the teacher, like, how's he doing? How's he doing? Is he causing you problems? I'll be the helicopter teacher mom. I feel bad for my friend in high school. His mom was a teacher, and she was that teacher that like everybody hated. And all they did was complain to her son. Like, why is your mom like this? Why is your mom like that? I hate your mom. I hate your mom. I felt so bad for him. No, he would be like, you're right. I hate her too most of the time, actually. Which made it even more sad. She was just really mean. Like sometimes I could be sarcastically mean, but hopefully you guys know I'm just being sarcastic. She would just be like actually mean and about stupid things. Like I remember this one girl, she was like, why are you wearing that? That doesn't even look good on you. Because two people are already gone the way. Okay, here is a phylogenetic tree. From a phylogenetic tree, you need to know what a node is. And you need to know that time moves in that particular direction. Oh, ignore the copy image, blah, blah, blah. That was for digital times. Nodes. So the node is kind of like the fork in the road, right? It's where two branches branch off. So at the end of a line, you have a species. Okay, so each of the ends, so if you look at the letters, letter G would be one species, letter B would be another species, letter C would be another species, right? Each of the ends of a line is a different species. When you go backwards in time, you can see, for example, F and I had a common ancestor about this long ago. Okay. F and E are very closely related because they have a very recent common ancestor. We would say F and E are more closely related species, not only because they have a more recent common ancestor, but we can also compare their DNA here. And if you look at the DNA between those two, there wouldn't be very many differences. You do not need to highlight every single difference of the letters. Okay, just notice that there are differences. They're actually kind of already highlighted for you, right? Okay. Okay, so you guys added time and you added nodes. I would make that a color. Color code the green circle, and color code your node definition. So you know this is a node. I know for sure there's a test question either on this test or an upcoming test on nodes. And what do they represent? They represent a common ancestor. And this is just giving us an example of how we see these molecular homologies. If you look at G and B, they don't even have any differences in this particular DNA sequence, right? Because they're so closely related. If you look at G versus I on the bottom, you have quite a few differences, right? So this is what we mean by molecular homologies. We can look at the DNA molecules and we can see that they are the same. And that is because they came from a similar common ancestor. Okay. We good? Cool. Here is an example of a phylogenetic tree showing you common ancestors of different species. Do I want to go to that slide? Yeah, we'll go. Okay. Did you know the human and chimpanzee genomes are almost identical, 99% the same? You guys knew that. Say hello to Michael Wigler. What a great last name. Okay, organisms share similar genes because they inherited them from common ancestors. So we can see on this that corn and Arabidopsis have a common ancestor about here on the tree. Look, people and Drosophila on this tree, they look pretty closely related. But this is a very basic tree that's not obviously including all of the organisms. This guy interested in genes involved in cancer, the human RAS gene. So smoke mutates this gene. And then you get cancer and it's bad. Okay. So he wanted to look at what does RAS usually do. We wanted to find out what normally does. So we looked for the same gene or homologue in yeast. If yeast have this RAS gene.
Middle school volleyball, you are now released for your game.
We could use yeast instead of people to study the gene's normal role. Okay, so use. Come on, you guys. So he's talking about homologs. Okay, so he found the homologue in the yeast. Doing this whole process. And down at the bottom, each of these clones contained a portion of the same gene, the yeast RAS gene. Sequence the gene, deduce its amino acid, and compared it to human. So as you can see here, we're looking at the comparison. Does it look pretty similar? Yeah, I'm finding a couple differences here and there, but it's pretty similar, right? So this would be an example of a molecular homology. Yeast and human have very similar DNA in this gene, the RAS gene. There's the differences. 1, 2, 3, 4, 5. It's not very many. We can also see the comparison of slime mold, Drosophila, and chicken. So look at humans are exactly the same as a chicken in terms of this gene.
Maybe I am a chicken.
Okay. Okay. So we saw that the two genes are structural homologs. Their amino acid sequences are very similar. Does not mean they do the same job. So they checked that out to see if it does the same thing. I think they found out that it did. Okay. Biotech stuff, biotech stuff. Yeast that incorporated the plasmid and its leucine survived. Okay, yeah, this is all biotech stuff. Ooh, fun. I think all I wanted from that was for you guys to see the DNA sequences. That was it. Here is just another example of a phylogenetic tree. These are all owls and eagles and falcons. You can just see from this that we can look at these phylogenetic trees, which were built to show us how similarly related things are. And again, these are being improved all of the time because of our improvements in the DNA comparison world. Okay, in terms of homework, 2 things. What do you think those 2 things are going to be? 3 things actually. Achieves. Syllabus statements. Nope, we're going to finish them tomorrow. Syllabus achieves and some sort of Google Sheets thing.
Oh, yeah. Oh, ours, right.
Your materials, you're ordering materials. Okay. Your achieves are coming up soon. They're due next week. So get those up. Also, I have assigned all of your achieves through February. So you can get all of those done now if you want. Right.